Okay, I'm going to start
Intense heat.
on Bergey's menu. In the year 1923, Dr. David Bergey, professor of bacteriology at the University of Pennsylvania, and his four colleagues published a Bergey's manual of determinative bacteriology and a classification of bacteria that could be used for the identification of many bacterial species. Bergey's and his team, they were the first one to give an idea about the classification system of the bacteria, and this book made a base for the references of the various different types of bacteria. And even today, the basis of this Bergey's manual is still used today. Okay, and there were various publications which were basically published till today. Okay, and the trust was made on the name of David Bergey. It is Bergey's trust, and the first manual which was published was named as Bergey's Manual of Determinative Bacteriology, and the last edition of the Bergey's Manual of Determinative Bacteriology was published in the year 1994. But despite its age and the contents, it still serves as a reference guide for the identification of bacteria based on physiological and morphological traits. And in the year 1984, the first edition of Bergey's Manual of Systematic Bacteriology was published. It contained descriptions of all bacteria and archaeal species which were identified till the year 1984. Now, Bergey's Manual of Systematic Bacteriology, the first edition, what the first edition contained, and it contained the kingdom Prokaryota, and there were four divisions. The division 1, which includes graciolutes; division 2, which includes firmicutes; division 3, which includes stenaricutes; and the division 4, which includes mendosicutes. And the division 1 basically graciolutes were the gram negative bacteria which possesses a thin cells, whereas in the division 2, which are firmicutes, they were the prokaryotes with the thick cells, indicating a gram positive type of cell wall. And the division 3, these are the prokaryotes of soft nature and lacking cell walls. And the division 4, which is mendosicutes, so prokaryotes having cell walls that lack peptidoglycan. In this manual, bacteria were classified into 33 groups called sections, and each bearing a common name rather than a formal taxonomic name. The sections are based on relatively easily observable characteristics such as shape, staining reactions, the presence or absence of cell wall, motility, reproduction by budding or by the mode of metabolism, and they contain both eubacterial and archaebacterial groups. So in addition to this, there are various other phenotypic characters which were used to divide this bacteria. But in the edition 1, these bacteria were not divided phylogenetically. That is the analysis of genes. In the addition to the reorganization based on phylogeny, the second edition of Bergey's Manual of Systematic Bacteriology possesses more information regarding the ecological information, of the each and individual taxa, and it is not grouped on the basis of clinical important bacteria as such, which first edition contained. Instead, all the pathogenic species are placed phylogenetically and thus scattered throughout the 5 volumes, and also includes phylogeny data. Now next comes the Bergey's Manual of Determinative Bacteriology. As I told you, 9th edition was the last edition of the Bergey's Manual of Determinative Bacteriology, and the arrangement of this group is basically strictly phenotypes. Okay, there was no classification based on genotypes. Okay, this was the limitation of Bergey's Manual of Determinative Bacteriology, but still it helps us to identify each and every taxa possible which are being discovered till then, to identify them. And the second edition, which today is being used, okay, up to last 2 or 3 years, okay, now a new edition is also come in the market, but with a different name. The second edition, which basically contains 5 volumes and was published over a number of years starting in the year 2001, and the last volume published was, I think it was around 2012. Okay, and the second edition, which is basically volume 1, which contains the Archaea and deeply branching and phototrophic bacteria, and in which 2 phyla of Archaea and 9 of deeply branching or 2 of the phototrophic bacteria are presented in this volume, and which was published in the year May 2001. And the volume 2, which is a single phylum, which is a Proteobacteria, which contains most of the gram-negative bacteria and represents largest phylogenetically coherent groups within the bacteria. Now the volume 3, a single phylum again, which is called as Firmicutes, contain most of the gram-positive bacteria with low DNA as well as the GC content. And volume 4, again a single phylum, the Actinobacteria, containing most of the gram-positive bacteria with a high DNA mole percent of GC. And the volume 5 and contains rest of the 9 phyla such as Planktomycetes, Chlamydia, Spirochaetes, Fibrobacteria, Acidobacteria, Bacterioids, Fusobacteria, Microbia, and Diacloglomi, which are gram-negative bacteria but are phylogenetically distinct from the Proteobacteria, have been treated separately. Okay, moving forward with the second edition. After the completion of the 5 volumes, okay, what did the second edition contain? Okay, so in this editions, total there were 2 domains, Archaea and Bacteria, and total there were 25 phyla, total 36 classes in this manual, subclasses 5, orders 89, suborders around 14, families around 203, genera 941 generas, and the species were a total of 5224 species. 5224 species. Now moving forward with the classification based on Bergey's manual. Okay, so what are the classes and how are they divided? Okay, as I discussed earlier, that the divided in 2 basic groups, that is, one of them is Archaea and the second one of them is Bacteria. Archaea contains extremophilic microorganisms. One of them is Craniarchaeota and the second one was Euarchaeota. Okay, and they contain Archaea, which differ greatly from the bacteria. And archaeal cell wall lacks peptidoglycan. Plasma membranes are made up of kinds of lipids other than the bacterial plasma membranes. RNA and ribosomal proteins are more like eukaryotes than that of bacteria, and some of the examples are Methanococcus, Thermus aquaticus, Thermoproteus, Halobacterium, etc., and there are variety of them. Okay, now with the classification of bacteria, one branch which is closely related to Archaea, that is Aquaceae, and this Aquaceae and Thermotogoa are the deepest or oldest branches of bacteria. Both are gram-negative thermophiles, and the Thermotogoa is named for its loose-fitting sheet or toga. Now moving further with its closely related family, okay, Thermotogoae, okay, it's I discussed right now, and the next one is of Chloroflexi, and next one is a Deinococcus thermus, and all these belongs to the group of thermophiles. Okay, all these group belong to the group of thermophiles. Okay, now moving forward with the gram positive bacteria, it contains low G plus C containing firmicutes and high G plus C containing actinobacteria, of which bacilli and clostridium are the common ones. Starting with the gram positive bacteria, largely solitary and many of them endospores, responsible for many significant human diseases including anthrax, some other like Clostridium botulinum, some are cocci which are also included in this group, and they cause common diseases, okay, such as food poisoning, and some of the examples are Staphylococcus, Lactococcus, Streptococcus, etc. Okay, so now moving forward with the second group, actinobacteria. These are basically some gram positive bacteria from branching filaments, and some produce spores and produce many commonly used antibiotics including streptomycin and tetracycline. And one of the most common types of soil bacteria which are also common in dental plaque, such as streptomyces, okay, even actinomycetes. Okay, now moving forward with the gram negative groups, okay, it may contain cyanobacteria, okay, or chlorobi, photosynthetic ones, spirochetes. Okay, now spirochetes are basically gram negative and common in aquatic environments, and rotation of internal filament produces a corkscrew like movement, and some spirochetes like Treponema pallidum causes syphilis, and the Borrelia causes a Lyme disease, which are the human pathogens. Now moving forward with the cyanobacteria and chlorobi, which are basically the photosynthetic bacteria. The cyanobacteria are photosynthetic bacteria common in both marine and freshwater environments, deeply pigmented and often responsible for the blooms in the polluted waters. Both colonial and solitary forms are common, and some filamentous forms have cells specialized for the nitrogen fixations. This is the only one of the main group of the bacteria which undergo oxygenic photosynthesis, and their mechanism is somewhat similar to that of plants, and their mechanism in which they use bacteriochlorophylls, not chlorophylls as in the case of plants. At the end of the process of the photosynthesis, they produce oxygen. Okay, whereas there are also other bacteria which do not produce oxygen at the end of photosynthesis. They are termed as anoxygenic photosynthesis. Okay, now moving forward with the group of Proteobacteria, okay, now beta ones, a nutritionally diverse group that includes soil bacteria such as lithotrophs, nitrosomonas, that recycle nitrogen within the ecosystems by oxidizing ammonia, and other members are heterotrophs and photoheterotrophs. Okay, so some of them may undergo photosynthesis but not the oxygenic photosynthesis, only the anoxygenic photosynthesis, which do not produce oxygen. Moving forward, the gamma group, A diverse group including photosynthetic sulfur ones. Some of them are pathogens such as Legionella and the enteric bacteria that inhibit animal intestines, such as the enteric one include E. coli, Escherichia coli, Salmonella, which cause disease, and Vibrio cholerae, Pseudomonas, which are commonly found in the soil and are responsible for other plant diseases, as well as some of them are opportunistic pathogens. And now moving forward with the delta group, the last one. Okay, these proteobacteria include bacteria used in the bioremediation such as geobacter species. In addition, this group includes predatory bacteria such as Dello vibrio and Myxobacteria, which are basically glider bacteria, which later glide in the multicellular groups and form upright structures called fruiting bodies. And Dello vibrio is one of the active predator of other bacteria such as Escherichia coli. Now the latest manual of Bergey's manual of systematics, okay, of Archaea and bacteria, okay, of Archaea and bacteria. It is the latest manual, Bergey's manual, which was published in the year 2016, and it works as a reference work aimed for the undergraduates and graduate students and researchers, professors, as well as other professionals. And this edition is a pure online edition. Okay, you need to simply search these keywords, Bergey's manual of systematics of Archaea and bacteria. You can go through the edition. Some of the part is free if you want to view. And the first edition contains a few hundred genera, around more than 600 plus species, which has been described per year for each of the last few years. Also contains the data of the old manual of Bergey's manual of systematics. Okay, that is second edition, first edition, even the data from the Bergey's manual of determinative bacteriology. And this tool provides an important information regarding the various bacteria, and it can be easily accessed because of the digital edition. And this digital edition provides up-to-date descriptions of the taxonomy, systematics, ecology, physiology, and other biological properties of all the prokaryotic taxa. Now given here, this is an actual photograph of the Bergey's manual of systematics of Archaea and bacteria. It was published in the year 2015. Okay, and this article is of Bergey's manual trust. Okay, if you want to view online, you can go through this DOI number. Okay, where a species is described. Okay, and in which family Berkeyoidariales order has been described. Okay, and you can clearly see that this bacteria Berkeyoidaria was basically analyzed for the phylogenetic analysis of 16S RNA sequences, and order is basically divided by phenotypically, metabolically, even ecologically, and includes information regarding the aerobic or anaerobic, chemoorganotrophy, obligates ones, or chemolithotrophy, or it may be nitrogen fixers, or its relation with animals, humans, plants, etc. Okay, and below given the Bergeyolderia, the name of the bacteria and the scientist which describe this species are given at the end of the reference. Okay, so I'm going to end the lecture. Okay.